because of the recent release of this FDA's roadmap map about how to reduce the animal testing from now on, and they have a 3 to 5 year plan. And so this actually triggered a lot of discussions in the industry, and from research part, from drug developer, from MNC, and all small biotech, and as well as actually affecting CRO. And people is wondering, how feasible this this proposed roadmap is, and what's the reality, and how do we deal with that? I'm here today actually to present this, actually to encourage more discussion, because everyone here, actually in industry, are waiting for FDA for more explanation and tell us what to do next. Yeah. So this is just a self disclaimer. I have 5 parts. First of all is the overview of the roadmap, and then spend a little bit of the time to talk about the NAMs itself, and the non-clinical assessments for antibodies and why they pick monoclonal antibody first. And for the first part, I just touch base about MS, multi-specific, bispecific, or even more specific antibodies, ADCs, and small molecules, to see how likely people are going to use NAMs for these drug development. Yeah, and finally is a summary. So first of all, the overview about this roadmap. Based on what they said, their goal is to have this 3R principle for animal testing, especially for the preclinical safety studies, while ensuring human safety. Yeah. So for key objectives, they said, well, first of all, promote development and adoption of the NAM, and using in vitro assays and also computational models. And they're going to enhance scientific validation and the regulatory acceptance of NAM, and also encouraging collaborations among stakeholders, industry, academia, as well as regulatory bodies. And the rationale, first of all, ethical reasons. And the second one is improved predictive power of the human relevant models. And lastly, to accelerate drug development and reduce cost. For the strategies, they say, well, first of all, need to invest in research and technology for those NAM methods, and to modernize the regulatory frameworks to accept non-animal data, and also increase the transparency and the data sharing to build confidence in new methods, because people usually think, animal is the best way to do the drug development, to find all the toxicities. Now you ask people to transfer to NAMs, a lot of people have doubts. So we need to build confidence. And for the expected outcomes, they expect to reduce the animal use in toxicology testing, and more efficient and human-relevant safety assessments, because when you use organoids, for example, you use human-derived materials to evaluate the safety. So you expect that those data will be more relevant than animal studies. Yeah. And also the strong alignment with global regulatory standards. Yeah. So the next step they have is, well, there's some ongoing validation of NAMs, and they are going to have their guidance updates from FDA to industry. And also they encourage stakeholder engagement through workshops and public forums. This is their action plan. So they want to initial focus will be on monoclonal antibodies, and begin with monoclonal antibodies, and then expand to other biological molecules, new chemical entities, and medical countermeasures. And in the next 3 years, they want to explore pre-existing international data, encourage sponsors to submit NAMs, and develop an open access repository with comprehensive collections of international drug toxicity data from animals to humans. So they want to combine all this using AI, use the machine learning to digest those, and The purpose, first of all, for the monoclonal antibodies, they want to reduce the routine 6 months primate toxicity testing for monoclonal antibodies that show no concerning signals in a 1-month study first, and plus the NAM test. And they want to cut this 6 months NHP studies to 3 months. So that's the initial goal. And they want to reduce the animal toxicity testing time frames and for other drug catalogs eventually. They want to change in toxicity testing will be tracked and quantified on a biannual basis. So they will discuss that and update. Yeah. And their long-term goal here is to, in 3 to 5 years, they're going to make animal studies the exceptional rather than the norm. So NAM toolbox, including human cell models plus computational models, will be the new standard, and animal study will be exceptional. So minimal animal testing considered if NAM is not enough. Yeah, but you need to provide a strong justification. Okay, so this is the action plan. So before going to the details, so you say why monoclonal antibodies is picked as the first one, I want to briefly touch on what are NAMs. And there's some types here. And the first of all is in silico part. The left part includes some computational modeling and read across methods. Here, I'm just showing some of them, like a QSAR and the PKPD modeling, and for ADMEs, and also in vitro and in vivo extrapolations, and also using the machine learning and artificial intelligence methods. And also the last one is the read across. So using the data from the same class, the compound from the same class, and to help us to make a decision. Yeah. So yeah, and the second one is in vitro. So organoids, all kinds of, for example, here human cell based. This is the advantage. 2D cultures, 3D cultures, and iPSCs, and here and also this last one here, tissue organ engineering, the organoids, MPS. Organ on chips or human on chips, yeah, etc. So the good thing is human related and also the organ specific, yeah. And last one here is some other texts, yeah. For example, those people are familiar with the omics, all kinds of omics, and also the imaging techniques, yeah, and also high-spread screening. If you want to use NAMs to make a decision, and there are so many biomarkers or some assays you need to go through, so you have they develop all kinds of high-spread screenings to help to make a decision, yeah. So where NAM have been adopted here right now for regulatory decisions, well, some I listed here is, for example, the skin corrosion irritation studies, eye damage irritation studies, that's skin sensitization and absorptions. So in FDA, actually, I helped to do for a couple of INDs. Actually, I initiated that and using the OECD approved test guidance. For example, here eye damage and skin sensitization for some rare disease products. So they haven't done the using animal studies to evaluate those things. So after extensive discussions, we think the OECD study is okay, is acceptable. So we allow that IND to go through a couple of INDs. Yeah. So here defined approaches. Yeah, combine in silico, in chemical, and in vitro data, and fix the interpretation procedures and integrate into regulatory framework. The performance. Versus animal tests, for example, here in vitro methods, you have to compare that and match or outperform, for example, here the local lymph node assay in specificity, and also validate for hazard identifications. So last here is some case studies here showing that for this is Captain and Forpet. This is some products for crop protections. So they did 18 in vitro tests, including skin, eye irritation, and airway toxicities. And they found that the NAMs actually, in combination of those tests, actually correctly identified irritants. and the risk assessments aligned with the traditional ways. So that's how they validate this and see and show that this OECD methods actually is okay to replace the traditional animal tests. Yeah, so this is the example I have here. Next here is hurdles. Why these NAMs hasn't been accelerated for adoption. So there's a couple of challenges here. And first of all, the challenge for complex toxicity endpoints. We all know that for repeat dose, for example, so for reprotox, for carcinogenicity, this is very hard. The endpoints are difficult to cover with NAMs. There are so many things you need to pay attention to. And NAMs must generate comparable data to animal studies. For example, the OECD, the 90-day toxicity studies. It's hard to collect all the NAMs and say, okay, if you use those NAMs, you don't need to do the animal test and 90-day toxicity studies. And the third one is need to cover all organs and systems and exposure durations and provide the quantitative point of departure. So make a threshold, like, departure from that threshold, up and low, that call as a toxicity finding. So this is the difficult part. Second one I'm going to talk about is the scientific and the validation challenges here. First of all, the NAMs provide the molecular and cellular readouts, but not direct apical effects. Okay. Second, you need a proof of predictive reliability via adverse outcome pathway networks. For example, here, the quantitative AOPS and in vitro and in vivo extrapolation to see the correlation. Yeah, so this is a lot of work. And the third one is complex NAM networks need to replicate full animal test data. So this is very hard. And lastly here, regulatory adaptation to complex NAM batteries is resource and time intensive. So it's very challenging. You want to finish all this in 5 years, for example. Cost is one thing, but bear in mind that if you have so many NAM technology, put them together, is this cheaper than animal study? Nobody knows. And lastly, regulatory landscape comparison. So the majority LAMs are not validated at an international level. So for big pharma or biotech, if they want to develop their products and for marketing approval in different countries, how do they make a decision? Yeah. For example, US EPA and FDA accept more NAMs defined approaches across chemicals and products. And Europe currently limits NAM use mostly to the one I just mentioned, the OECD skin sensitization and eye damage guidelines. So how about other countries? So a biotech companies, they need to make a decision. Okay, if FDA said, well, NAMs is good enough, but the other, like EMA doesn't allow that, what should you do? You may end up still need to do the animal test. So it's a battle, and I think a lot of communications for the intent internationally. How do we validate that, set a standard? Yeah, so everybody can recognize that. So back to the antibodies, monoclonal antibodies. Here, this is ICH-SX. So if you want to develop antibodies for non-clinical part, so I just highlight, I'm not going to go through all the details, but the highlighted in yellow are the parts that you need animal to do the study, to make an assessment. For example, here, first of all, is in vivo PD effects. That's for the efficacy study. Definitely, you need animal models, okay, in a relevant species. And then for PK/TK studies, well, you need the dose and frequency. You need to find that. You need to determine the exposure and what's the correlation with PD. And you might need to do a PK/PD modeling. And the last one I'm showing here is the anti-drug antibodies. That's a debate, and we agree that this is, the data from animal studies, even NHP may not be relevant. So that's one of the major limitations. And going to the general toxicity here, single dose tox, repeated dose tox, you need animal work. Yeah. And lastly here, reprotox, sometimes depends. So sometimes you will need animal studies as well. Yeah. So challenging here for monoclonal antibodies when you use the animal models. First of all, here is the species-specific immune differences. This will limit the predictivity. Yeah, the immune system is variable, and the human and animal actually have some big differences. And also the impact on drug response, the target binding affinity, the selectivities are different. So you need to be very careful when you interpret the difference. Yeah, and last one here is immune activation and toxicity. So it can be misleading due to the low or high sensitivity or irrelevant immune response. Yeah. The second one here is immunogenicity in animals often doesn't translate to humans. ADA, I just mentioned that. Usually when we develop a monoclonal antibody, it may be humanized or even is chimeric. And those proteins, well, in animal, they were recognized as a foreign protein. So likely they're gonna, depends on how strong the, so they can, have a high reaction and you see a lot of incidents of ADA, and sometimes it's weak and transient. So it all depends. So it has a very poor predictive values, and even you have ADA, it doesn't necessarily translate it to an effect on EK exposure or in other toxicities found in animals. Yeah. So there's one example of TGN 1412 in that roadmap. In the early days, actually, people doesn't have an idea about where this CD28 receptor is. Actually, the one actually causing the cytokine release syndrome is from the CD4 plus effector memory T cells. So eventually, they found that in NHP, actually, it doesn't express these CD28 receptors. So that's why in monkeys, they never pick up these toxicities and causing the tragedy. Yeah. So why MAP is first listed in this FDA roadmap? Well, potential reasons here. First of all, monoclonal antibodies compared to other modality is kind of relatively simple because it's very specific. You do go through all the screening. You find out this product for toxicity. There's the reason. So there's a clear mechanism of action, and it has a very well-characterized safety profiles. You basically can know based on the target you're working on, and reduced complexity compared to other modalities. For example, small molecules, that's very complicated. And the goal here, well, according to FDA, they want to use NAM test to shorten the 6 months NHP toxicity study to 3 months. Yeah. Based on experience I have in FDA, when I review those monoclonal antibodies, and also based on literature, for the non-PD related toxicities in NHPs, there are two. One is immune-related effects, for example, infusion reactions, cytokine release, and second, anti-drug antibodies formation affecting the PK. So you have a lower exposure, so decreased drug efficacy. Yeah, seen. Yeah, and the second one is organ-specific toxicity. Actually, is related to, for example, immune complex deposition. When the ADA is formed, they're going to bind to the drug and then deposit in certain organs, for example, in kidney and in vessels. So there's some specific toxicities you're gonna see, yeah, as well as some liver enzyme elevation, hepatocyte changes, yeah. So clinical relevance to these NHP studies, and here I see that first of all, acute immune reactions also noted in patients, and this is so if you shorten the 6 months to 3 months, that's not a problem. Usually, when you have the first injections and the second injections, you will see that, and so that can be picked up. And second of all, some chronic kidney and vascular toxicities rarely reported in patients. And lastly, the ADA affect the PK noted in some antibodies. So so there's some nice correlations, but not all. And conclusion here is the goal might be possible. I mean, from 6 to 3 months, because most of the toxicities actually can be picked up in early phase, but some of them may not, and some may not be relevant. So for example, ADA formation, sometimes it only show up in 3 months or 4 months or 5 months for transient times. But these are not considered a very important findings. And also in clinic, even ADA is fine. It can be treated or prevented in advance. It's reversible. So that's why I say might be possible, but it determined case by case. The exact name needed, there's some in vitro studies can do, and when you do the drug design, and you can use some algorithm to make some prediction. But how well are those how reliable are these algorithms is still a question mark. So multi-specific antibodies and ADCs, well, I just showed that here. Compared to monoclonal antibodies, multi-specific antibodies and ADCs are much more complicated because you're adding, for example, multi-specific antibodies, you're targeting 2 targets or even 3 targets. It's much more complicated. And also for ADCs, you introduce the payload, and the payload can be released to systematically. So it adds a lot of complexities here. So I think monoclonal antibodies would be the first to pick if you want to follow this road. Yeah. And for small molecules, it's much more complicated because usually small molecules are not that greatly specific. There are a lot of off-target toxicities, and their metabolites, they have broad distribution. So that makes it much more complicated and challenging. Yeah. So in summary here, NAMs are great tools for better human health protections, but there are a lot of challenges here. And we still need to evaluate how good they can mimic systemic, chronic, or multi-mechanism toxicities like carcinogenicity or reprotox. Yeah. And validation of NAM tools can be a continuous effort for new drugs. Even we have, for the same purpose, we use different cells. So which one is relevant, which one is not? We never know. And to gain international acceptance will take time. For monoclonal antibodies, shorten a 6-month studies to 3 months might be possible, however, requiring extensive data mining and careful and comprehensive analysis together with or human data possible. And then for small molecules, etc., they're much more complicated than a monoclonal antibody. So I did get some questions from the field, and I just picked some of them here, and it reflects the opinions from the field, but it's not full. For example, here it says that people told me that to make animal studies exceptional rather than the norm for safety assessment is in 3 to 5 years is a very tough goal. And a stepwise approach is needed, and for quite a long time, NAMs and animal studies should coexist. And the third one, NAM and animal studies, which one dominate? It should be case by case. And I think for a long time, animal studies will still be needed. And the NAM, in general belief, it can help to explain why there's a toxicities. And then for future, for the same class, we're going to do a screening first to avoid such kind of toxicities. Yeah, clearly more discussion are needed. So that's all. Thank you.
Thank you very much, Dr. Chen, for your excellent summary for the FDA new policy for obesity testing. We will save all the questions to the end. So we will transition to our speaker. Thank you. So our second speaker is Dr. Frank Zhong from Glycosite-Specific. Dr. Zhong is the director of medicinal chemistry at Glycosite, and he leads the development of Glycosite-Specific Antibody-Drug Conjugate platform. With over 15 years of experience in organic synthesis and drug discovery, he has also the modern physics peer-reviewed publication. Trained as a carbohydrate chemist, Dr. Zhong advanced his expertise in chemistry and the chemical performance. During his first work at the University of Arkansas, since 2018, he joined the WRC1 group at UNG to work on antibody glycan remodeling and developing novel glycosite-specific ADC platform. Prior to joining Glycosite, Dr. Zhong as a faculty at UNG.
Thank you, Dr. Chen, for the kind introduction. And I also would like to thank CBA to give me this opportunity to speak here. Today, I will talk about glycan remodeling of antibody and its application in the development of glycan-specific antibody-drug conjugate. So, So conventional chemotherapy has been used as a major treatment for cancer over 70 years. However, chemotherapy has a major disadvantage. So the selectivity between normal cell and the cancer cell is poor, which can cause side effects. While antibody-drug conjugates take the advantage of the highly specificity of an antibody, it can deliver cytotoxic drug to the biomarker expressing cancer cell and internalize the toxic drug to the cell, then release the drug, eventually causes the cell death. So it is very rapidly developing, and up to date, there are 18 ADCs already approved by FDA and other drug regulatory agencies. So how to make a ADC molecule? So there are four key components of an ADC. So first is antibody. So so the antibody takes the, it's responsible for the target delivery. And another one is the payload, which is responsible for the cell killing. And the linker, which can connect these two. Another key component of ADC is how you conjugate the linker payload to the antibody. That's the conjugation chemistry. There are many methods for the antibody conjugation. The early generation of ADC rely on like lysine or cysteine conjugation. Because there are a lot of lysine in the antibody, so this kind of random conjugation generate heterogeneous ADCs, which is like a problem in the quality control of during the CMC process, and also may cause stability or side effects. So then the next generation for the ADC, that is the site-specific. So in HER2 is a homogeneous ADC with a DAR number of 8. And the conjugation is through the interchain cysteines, because there are totally 8 cysteine, interchain cysteine, so they can get a homogeneous DAR 8. But this is just a special case. So there are still challenges to use the cysteine for the conjugation to get different DAR numbers. And the other site-specific conjugation methods, including engineering cysteine, also this technology called CellMab. Or you can incorporate unnatural amino acids which carry bioorthogonal tags in the unnatural amino acid. Or you introduce fusion proteins, or use the enzymatic coupling method. Another big category of the site-specific conjugation, that's the Fc glycan site-specific conjugation. So actually, all the human IgGs have a conserved Fc glycan at the asparagine 297 position. So there are two features of this Fc glycan. So first, it's very heterogeneous. When you express an antibody, so it carry like a mixture of different glycans. But this glycans has a profound effect on the effector functions, such as antibody-dependent cellular cytotoxicity or ADCC. For example, For example, the co-fuel cosylation and the terminal sialylation, it can decrease the ADCC. But the galactose, it can enhance the complement-dependent cytotoxicity or CDC. So the terminal sialylation also found it can increase the anti-inflammatory activity of IVIG. So because this Fc glycosylation is very important for optimal the therapeutic efficacy. So to remodel this kind of glycosylation, we will have a potential therapeutic effects. So how to do glycan remodeling? One way is you use like engineered cell to during the process, during the expression procedure, you control the glycosylation. Another way is you don't have to engineer the cells. You just get any wild type antibody. You use some enzymes to do the glycan remodeling. Dr. Lai Wang's group at the University of Maryland, so is a leading pioneer in this kind of glycan remodeling field. The process is usually two-step process. So the first step is you use endoglycosides to cleave the heterogeneous glycan between the glycosylation bond between these two gluconide. And then the second step, use endoglycosynthesis mutant to transfer structure well-defined glycan back to the antibody. Dr. Wang's group have developed the different endoglycosynthesis mutants for the transference of different glycan structures. Because this kind of glycan remodeling method is so useful to enhance the therapeutic efficacy. So Dr. Wang found a company, our company, GlycoT Therapeutics, to further develop this technology. So here I will show some examples how we do glycan remodeling of monoclonal antibody. So rituximab is a monoclonal antibody for the treatment of blood cancer. So as I mentioned earlier, this co-fucus will decrease the ADCC. So in order to improve the therapeutic efficacy, we want to remove this kind of co-fucus. So there are two-step conversion. First, still you use a wild-type enzyme endo-S2 and alpha-fucosidase to remove the original glycan. And the second step is you use a mutant to transfer a G2 glycoform to the rituximab, and then you can get the defucosylated G2 rituximab. So this glycan remodeled rituximab, it shows like this is the LC-MS data for the remodeled rituximab. You can see here. So before remodeling, it has a heterogeneous glycan, but after remodeling, you got a homogeneous glycan. And this remodeled rituximab has significantly increased the affinity to Fc gamma receptor. Fc gamma receptor is responsible for the ADCC effect. So also the ADCC assay showed this remodeled rituximab enhanced the ADCC by over 50-fold. So then through this glycan remodeling method, we can get a so-called bio-better rituximab. Another example is glycan remodeling of IVIG. IVIG is a mixture of IgGs, which is important, which was used for the treatment of autoimmune diseases. And it has anti-inflammatory activity. So one major issue of IVIG treatment is the high dose. Actually, you have to use 1 to 2 grams per kg, this dosage. Actually, the sialylation in the IVIG was found it's responsible for the anti-inflammatory activity. However, in the IVIG, the sialylation level is pretty low. It's below 10%. So then we use the glycan remodeling technology through a two-step conversion. We can introduce this high sialylation glycan to the IVIG. So then the preliminary data of animal study, it shows like the anti-inflammatory activity increased over 10-fold. Then it can significantly reduce the dosage. So using this glycan remodeling technology, we can also make a glycosite-specific ADC. So the second step, when we introduce a structure well-defined glycan, if we introduce a bioorthogonal handle or tag to the glycan, and then use an enzyme to transfer to the antibody, then we will get the AZ tagged antibody. So this slide shows how it works. So first, you get a wild-type monoclonal antibody, and then you use a wild-type enzyme called endo-S2 to cleave the heterogeneous glycan. And the second step, you introduce the AZ tagged glycan back to the antibody, get the AZ tagged antibody. And then the third step is you use a click chemistry to introduce the drug payload on. And based on the structure of this of this glycan, so we can generate ADCs with different AZ based on the different structure of this glycan. So this is Glycote's first generation of antibody-drug conjugate platform. So we licensed this technology to Daiichi Sankyo in, I believe, 2020. And then it went to the clinical phase 1 clinical trial in 2022. And late last year, they reported a promising phase 1 clinical data. So this method is pretty efficient, but there's still a drawback. That's because to prepare this glycan, you have to isolate from the chicken egg yolks, and the yield is pretty low. So it might be a limitation in the later stages CMC. So to solve this problem, Glycote's second generation of ADC, we try to simplify this glycan structure to just a disaccharide, and then try to use this simplified disaccharide do the glycan remodeling. So this disaccharide can be easily synthesized from the monosaccharides, and the synthetic route is flexible. You can introduce different number of the AZ tag. So then remember previously we used a wild type enzyme to cleave the heterogeneous glycans in an antibody. So here we found like this enzyme will cleave the original glycan, but simultaneously it can also transfer this disaccharide structure to the antibody. So that means compared to the first generation of technology, here we just use only one enzyme. Just one step, we will get the AZ tagged antibody. And this method is very efficient. You can see here, we just only need like 0.1% by weight of the enzyme. And you just treat 30 minutes, you will get almost complete glycan remodeling. And for the structures with more AZ, we just increase the usage of the enzyme and extend the reaction time. So eventually, it can also get a homogeneous antibody. So after get this AZ tagged antibody, the next step is you introduce the linker payload through click chemistry, and we can generate antibody-drug conjugates with different DAR numbers. And this is the IOC mass analysis of the synthetic ADCs. It shows the quantitative conjugation. So I need to point out here, this peak is not the unconjugated antibody, it's just the fragments during the mass analysis. So the in vitro study, it shows this ADCs, it has a dose-dependent killing for the HER2 overexpressed cell line, BT474. And the ADCs with a higher number, it shows better cell killing. And for this HER2 low expressed cell line, and so when the DAR number is low, it don't have any killing. But when the DAR is pretty high, it showed some off-target cytotoxicity. Mainly it's because of the payload is very hydrophobic, and then it may adhere to the cell membrane of some other cells. So then cause the off-target cytotoxicity. So using the glycan remodeling technology, we generated the first generation of our ADC and a second generation. So it is pretty stable in the red serum and also have low aggregation. So, and also it didn't change the affinity to FC RN receptor, which is responsible for the stability. The xenograph mouse model, it shows like this exhibited the tumor growth is significantly was reduced by just one single dose injection. And we also tested some other substrates, just like so the original technology is used the disaccharide which contain mannose. So and then we also tested the disaccharide with glucose and galactose. For the activity, the mannose is the best. The glucose is also pretty good. The galactose is the worst. So, but from the synthesis, the glucose disaccharide is much more easier to be synthesized. So we also used this disaccharide for further development to make our ADCs. So if we preload the drug to the disaccharide, then we can use the similar method, to use a wild type enzyme and then cleave the original sugar and simultaneously transfer this drug preloaded disaccharide to the antibody. So through this way, we can just use one enzyme and a single step from the any wild type IgG antibody, one step get the ADC. So this method have big advantages. First, of course, it's site specific. Second is it doesn't need to perform the genetic engineering of antibody, and just use one enzyme and one step. It's very efficient. So besides the IgG1 we tested, it also works on other types of IgGs, including IgG1, 2, 3, and 4. And it also tolerates different linkers and payloads. Besides the payloads, we also try to introduce the other ligands. It also works pretty well. So here, what I want to mention is like I believe antibody work in the site-specific antibody drug conjugate should know Sinofix technology. So because several ADCs have already go to the clinical stage, especially the Innovent IBI 343, it's already go to the phase 3 clinical trial. So their technology used the two enzyme, used one enzyme to cleave the heterogeneous glycan, and then they use a second enzyme to transfer the UDP GalNAc to the antibody, get the AZ tagged antibody. And then they use a third step to click the cytotoxic drug onto the antibody. So this is a two enzyme, three step stage. So well, Galactose technology is you just either you can use like a one enzyme, two-step conversion, or you can use just the one enzyme, one-step conversion. You can just get the ADC, and it's very homogeneous, and so then Innovent went to us. For their next generation of ADC, they have already licensed our technology. So here is a summary. So Galactose currently have two platform technology. The first one is chemoenzymatic glycan remodeling of therapeutic antibodies to enhance the antibody efficacy. Second technology platform is use the single enzyme, single step to generate glycosite-specific homogeneous ADC. So if anyone have interest to this glycosite-specific ADC technology, we are very glad to help you in your ADC development. Thank you very much.
Dr. Xi Yang. Dr. Yang is a former FDA master reviewer and a board certified toxicologist. working as a senior toxicologist at RTI. She provides guidance on regulatory support, strategy, and the submission for drug development projects. With over 15 years at FDA and with experience, Dr. Yang has expertise in evaluating drug safety, ranging from IND to NDA and to post-marketing. She has published more than 50 peer-reviewed publications and book chapters. Dr. Yang, please.
Thank you. Thank you so much for the meeting organizer and especially Dr. Xinrong Chen and Yongbin Zhang for inviting me to share my personal experience with NAMs and how I renew this trend to having these NAMs into regulatory decisions. So here is an outline for my talk. First, I'm going to tell you a story to show what's my impression about these NAMs and what we can take from the story, since many wisdom is hiding in the story. And then I'm going to go back to show, so why do we need a non-clinical study? Why do we need to NAMs to replacing the animals? What are the limitations of the current approach and how NAMs can help us to create a more innovative solution to make the drug development cheaper and better? At last, it's the future, how it may look like and how we can get there. So you may heard of this story. It's Sorcerer's Apprentice. It's coming from a very old German story, but then make a world famous by this Disney short film. So Mickey here, he is the apprentice, an intern of a very powerful sorcerer. And his daily job is to transferring a bucket of water from one well to the other well. And he found it's very tedious. It's not a good job for him. And his boss, the sorcerer, is very powerful with the magician power. So one day when his boss is not at home, Mickey put on his magic hat and then turned a broom into like a robot to do his job. So now since the broom can do the transferring of the water, Mickey fell asleep, and in his dream he become very powerful, do a lot of great things for the world. However, when he wake up, he just discovered that because this broom didn't know when to stop, so he keeps transferring this water from one well to the other. So the whole house was flooded with water. So Mickey was so scared. He didn't know what to do. So he just used a axle to chop the broom into the small splinters. But it didn't stop the broom. It just turned out to be hundreds, thousands of broom keep doing the same job, transferring the water from one place to the other. So the situation got worse. However, finally, this powerful sorcerer came back, and even without this magic hat, he just make it disappear and save Mickey. So my take-home message from this story is that when a new technology come into the world, it's better to know what it can do, what it couldn't do, than before you move it to do the task you are supposed to do. You may hear that Dr. Chen gave a very good presentation about what's this roadmap, what does it mean, and how this has been created a shock in the field. Actually, I want to also add a little background about that. The NAMs that as earlier as in the 2007, there is already a Tox-21, which is collaboration between all the government, federal governments like EPA, FDA, NIH. They have been coming together to talk about what will be the toxicology testing in the 21st century. And then there is the OECD workshop, and there's a typo here, 2022, the Congress passed the FDA Modernization Act, and that also caused some discussion during that time. However, I want to highlight one point is that all this doesn't mean it will end the animal testing, at least for now. Even for right now, the phasing out animal, they will start from the relatively not so much rely on the animal studies, for example, the antibodies. They just want to give another options for the toxicology testing and to improve the situation that the one animal couldn't be the best solution. So now I'm going to just take a step back and show why do we ask for the non-clinical study, and then we can continue the discussion how NAMs will help us to address those questions. So the non-clinical studies are designed mainly the purpose to predict the human safety and efficacy, especially when there's a first-in-human study. If you do not have any experience with these new drug candidates, how are you going to choose the dose? And how are you going to make sure sometimes those are the healthy volunteers won't have suffered from those new drug candidates? So as I mentioned here, the NAMs, which is no observed adverse effect level, can be very important to drive the first-in-human safe starting dose. Sometimes if for different indications, other methods can also be used. So that's the limitation of the NAMs, how we are going to translate the NAMs finding to the human. Sometimes the animal study, the results will also help to determine a safe high dose. For example, oncology drugs, sometimes they were associated with very severe cytotoxicities, and how to choose the stopping rule, how to choose the highest dose that can also be collected from the information gathered from the animal study. And then also important to address some other potential toxicities that couldn't be identified using the clinical trial. For example, you don't want to see the teratogenicity to the harm to the fetus when you give the drug to the pregnant women in the clinical trial. So you prefer to have those knowledges earlier. And we all need to know that all the animal study results will be represented in the drug labeling. And the drug labeling also sometimes will influence the drug price. So that's why during the drug labeling negotiation, there's also multiple rounds of discussion about how these results be presented in the label. If you have been working with the regulatory affairs agents to put on this IND application, the submission, you will know that there are many different kinds of toxicology studies will be in the module 4, and there will be the 2.4, 2.6 summary documents to summarize those animal studies or sometimes in vitro studies. And my point for this slide is that yes, it's true that many studies have been conducted in the animal, but there are also many in vitro studies being part of this IND submission package, which means some of the technologies, some of the assays have already been used in the regulatory decision. For example, here I want to highlight the secondary pharmacology. As Dr. Chen mentioned that for the antibody, it's have a better off-target profile, so you do not need to worry too much. But for the small molecules, they can be designed promiscuous. Therefore, they can have many off-target effects, and those off-target effects sometimes will cause the toxicity. And how did they do that to prevent and to get prediction about off-target? They use this called the in vitro screening assay for the secondary pharmacology to know what are the potential targets will be hit by these small molecules. And for the sometimes for the biologics products, that you can use the tissue cross-reactivity TCR assays to predict what kind of off-targets will be there. And the timing of the non-clinical study will also be important for the whole drug discovery development, because if you have you are working on a very hot target and you have many competitors, you really want to move your programs ahead as fast as possible. But you also have limited resource in the very earlier stage. Therefore, you need to think about when should you have your studies conduct, when should you have the synthesis data sets ready. Therefore, you won't put your clinical trials on hold just because you didn't have a good planning in the earlier beginning. So the timing normally is for the earlier pharmacology studies, they are generally done in the even parallel with the discovery phase, primary pharmacology, in vitro, in vivo. And then for the genotox study, they normally should be done before the phase 2. However, many sponsors, they choose to complete all this in vitro, in vivo genotoxicity studies even during the pre-IND stage. And the PK information normally should be available, especially for the drug-drug interaction, the major metabolites, they need to be characterized. And also, importantly, for the embryo-fetal developmental talks, they need to be finished before the phase 3. So many companies, they choose to conduct their fertility study, embryo-fetal studies in parallel with the phase 2 clinical trial. Dr. Chen already gave a very good introduction about the NAMs and also talk about that how NAMs can help and cannot help with the animal study. So here I'm trying to also summarize what are the promising areas associated with the NAMs and how they can help with the animal approaches. So for example, for many NAMs, they are using the induced pluripotency stem cell derived cells, for example, heart cells, liver cells, kidney cells, lung cells. And since they are derived from the human original cells, then they theoretically will be better than the animal study to reflect how the human body will be react with the drug candidates. And for the throughput, we know that for the animal study, you can limit it by how many animals you can use, since you don't want to use too many animals. However, for NAMs, the throughput will be better since you can have the different platform like the 96 wells, 384 wells, or even higher throughput. And for the animal study, I sometimes hear another criticize is that you non-clinical people didn't do a hypothesis-driven. You just observe what's happening after you give the animal the testing drug. So that's true. And so hopefully in the future for the NAMs, we can use this system to test a hypothesis instead of just observe what's been happening. And the study duration normally is shorter with the NAMs, but that may not be a good thing because we know that some of the drugs will be used for a long time. Then how can you convince that the regulatory, or you can convince yourself that a 2-week study will be long enough to pick up all that the potential adverse reactions? And at last is the operation expenses, because people really want to get a better drug to the hospital for the patients using less resource. But we know that animal study can be very expensive. Right now, is for the monkey study, it can be millions of dollars to spend on one study. It's true that the NAMs can have a lower price, but we know that currently the NAMs are only have especially for in vitro NAMs, it can be only one organs, liver or heart. If you multiple them together, it's true that it's hard to predict which one is better. So this I want to show this picture is because it's a roadmap for the NAMs in the drug development, and also it can be applied to many new technologies or even for the AI, since recently FDA announced that they're going to use the AI in their drug reviewing process. So how to move the new technology from the earlier beginning to the FDA regulatory acceptance, that's need a lot of communication, collaboration, like this earlier this morning, multiple speakers mentioned that we need a kind of get together like this meeting at here to share ideas, to inspire the innovations. Because sometimes at different stage, what you are asking for, what you're wishing for may be different. For example, in the earlier drug discovery stage, those NAMs will not need to be qualified. as long as it shows that they can be repeatedly pick up the signal, they can be used as a tool to help the company to choose their lead compounds. However, during the FDA reviewing stage, it's better to have many solid evidence to prove that those assays, those names have been very well characterized. You know what kind of endpoints it can be used and how these endpoints can be linked with the human responses. Because when I was doing my postdoc at NCTR, I have been doing some biomarker works, and I want to share some lessons learned from the biomarker work. Since in the earlier 2000s, there are thousands of biomarkers has been published every year, and how they change this the new technology then become a very powerful tool to help with the oncology drug and patient prediction, response prediction. It's also a very long process. So like the book I recently read is that when we went back to look at the history, we only remember what happened when the new dynasty has been created, been established, but we forgot there's a very long history before that to prepare these new things to be there. So similarly for the biomarkers, it went a long way together for the public-private partnership to get together to identify what are the needs, what are the gaps there. So they find that you need a very standardized terminology to define the classes of biomarker. It can be biomarkers for the safety, it can be biomarkers for the prognostic, but it's better for you to understand what kind of situation your biomarker can be used and in which specific questions they can address. So it's need to be a narrow the context of use. And then you have that tools to develop for the fit of purpose. However, the good thing about this non-process that once it's qualified, this method can be used without additional demonstration. And now I'm going to use going deeper to use specific cardiac names to show how these names can be used for the regulatory applications. You may heard that for the human-induced pluripotent stem cell-derived cardiomyocytes, that's cells that can be beat in the cell culture wells, or they can be used to generate the organ, the chip, heart on the chip. And this new technology was first commercialized in the 2012, and about that 3 years, 2009, and then 3 years later, it's been showing up in this R&D stage, since people have been trying to use these in vitro tools to answer some questions that couldn't be answered in the animal studies. First, because people want to use this in vitro cells to recapture the different ion channel activities in humans, because at that time the counterpart for this in vitro assay is a hERG assay. So hERG assay is only focusing one ion channel instead of a comprehensive ion channels. And later, the use of this in vitro technologies has been broadened to not only the calcium, the ion channel or sodium channel, but more other structural damages to the heart, the cardiac myocytes themselves. And then later, we know that normally if you want to push a new technology or new regulatory pathway, it's better to have a guidance. So that's how this NAMS has been following. They in the 2022, they have this SHE14 S7B Q&A to try to show how the FDA, how the regulatory agencies think that what kind of best practice you need to have a new technology, this NAMS, to be support the regulatory applications. And here is another case I want to share is that in certain situations, the animal models will not give the best answer than how to use the NAMS, the cardiac myocytes, to answer a question. So here, when the drug compound have been tested in the animal study, the cardiomyofiber necrosis was found in the repeated dose studies. And another thing about this drug candidate is that they have a different metabolite to parent ratio in animals versus in the animal species. In human, there are more parent. So the metabolite is about 10% of the parent. However, in the tested animal species, there are more metabolites to the parent. And we speculated that this metabolites itself will contribute more and leading to the drug-induced cardiotoxicity. But because the animal, the human, they have a different metabolite to parent ratio, how are we going to confirm that it is the metabolite driven to that result? So what the proposed approach is to using the iPS-derived human cardiomyocytes and then treated the cells with either the metabolite itself or treated with the parent compound, and then compare to that IC50 and to see that here it's in this human relevant model, even it's in vitro, but we did show that the metabolites have been driven the cardiotoxicity. But the question is how we translate this in vitro finding into the human clinical trial. Is that true that we can just directly use a factor to contribute to the metabolite to parent ratio and through the AUC and to persuade the FDA that we want to go higher dose, since in the human there is only 10% of the metabolite, there is a the metabolite to parent ratio in the dogs, in the rats are different. So there are many more questions need to be answered before we really go there to the FDA. So I'm going to skip this one since many of the limitations and the barriers has been discussed at my current presentation and also by Dr. Chen. The take-home message is that if the NAMs are going to be support the regulatory decision, especially for standalone assay, for the safety decision, the bar is really high. And as we show that in the biomarker example, how are we going to move new technology from the early beginning to gradually into the regular decision? We need a lot of data. For example, in the current situation, when we use the NOAEL to drive the human safe starting dose, we use a factor of 10. Why not 20? Why not 5? Those are built on the scientific data. So in order for the NAMs to be incorporated in the regulatory decision, we also need a lot of data. We need a collaboration. So sometimes the company will suggest the FDA or the other consortium to create a safe harbor for them to share the data and therefore to help them to build the models better. So that's the last one of my presentation. We know that we are very enthusiastic about these NAMs since there are limitations of the current approach. I also have confidence that the field will go to a better future, since for the science it's always self-correcting. In the very earlier beginning, there's no animal study required for the new drug, and gradually we have the animal study. We know that there are limitations of the animal study, so we have different in vitro assays written in those different guidance. And in the future, I believe this innovation, the new tools could help to move this field more efficient. So with that, I'm going to close my presentation, and I believe next we are going to have a panel discussion for any questions.
For the family report, we have about 50 minutes for questions. I want to ask, do we have any questions from online audience? So we will start with the audience in the room. Do we have any questions from the room?
Thank you for the very nice presentation. These questions for the last speaker. So you mentioned for the iPS cell-derived cardiomyocyte and like other, like this. Like other these cell systems, usually they don't contain a liver metabolism capability, right? So are there, like more multi-organ integrated co-culture system can solve that problem?
Thank you so much. That's a very good question. Yes, it's true that many of the drugs, they need to be metabolized and they need to be using the liver, pass through the liver to be functional. And for right now, and my knowledge is that some people are trying to link this in vitro liver chip with the heart chip together, therefore to give a more predictable situation in the human. And the challenge is very challenging since we know that for the liver cells, they have their own liver cell culture medium, and then for the heart, they use a different cell culture medium system. How do you want to make sure that you need to have a same cell culture medium to make sure that they can pass through and the drugs can be slowly, gradually go from the liver to the heart, and we all know that it's not only liver and heart, there are other kind of organ systems. So it's a very challenging to put them together.
Thank you.
Do we have any other question from the audience or from the panel speaker?
Thank you for a great talk. I missed probably one of the first talk. So I do have a question for any of you, maybe. Yeah, so we do notice that recently there was a kind of like a regulatory agency's announcement, right? So we saw the news. So they're trying to replace some of the preclinical data, preclinical experiment or animal experiment with the organoid. So I wonder, like any of you, whether you can share your feedback on that. I know it's a hot topic, but feel free to share your, personal insights or your comments. Yeah.
You mean the, when they want to ask the companies, maybe they need to use less animal studies and also use more NAM, right? Yeah. Is that?
Yeah. I don't know whether you saw all the media, like we saw that kind of like the announcement or the news. So they're trying to replace some of the preclinical data, some of the preclinical experiments with an organoid experiment.
Yeah, I think that the roadmap also talking about, yes, they encourage sponsors to provide both data for them to compare, right?
Yeah.
To validate. I think it's going to take a while, and maybe they have some incentive to ask, when companies provide that, they can, the FDA will give you more close or frequent communications. That's why they encourage you to do that. But I think it's going to take a time. When you have those data with existing data, if FDA already have, how do they decide, whether this NAM studies is enough or not? I think I have no clue yet. I think it also depends, the NAM studies, assays they're using, and is it validated or not, to what extent, and also fit for purpose. I think even, I work in CRO now, and companies asked us, should we do some NAM studies? And I say, we don't know. And yeah, so I really don't have a good answer. Yeah.
Yeah, great being said. It's a very challenging, especially for the safety decision. There's a lot. So, for example, I want to share that one of the guidance for the embryo fetal developmental study, when you can use in vitro assays to replace, the situation is that if for this class of compounds, they've already been shown in the pathogenicity, then if you have a second drug candidate in the same compounds, the same drug class, you can use that in vitro technology to say, oh, okay, our compound have the same concern for the carcinogenicity, then you do not need to repeat the animal study and say, okay, those compounds will cause the fetus dysfunction. In those such situations, you can use the in vitro to confirm that we do not want to waste the animal, we want to reduce the use of the animal. So we can work on those low hanging fruit first, and then gradually move to the other part of the situation.
Yeah, I'll add a little bit. I think, if it's starting from scratch, it's very hard to make a decision. If you're working on a totally new modality or total new antibodies using a new payload, there's a lot of uncertainties there. But for something already known, or for example, for the same class, we already know based on mechanism, there's a potential for carcinogenicity, for example. Then there's no point. There's a weight of evidence studies. You can justify that. Okay, I'm going to put in the label, say, there's a potential. Yeah, but you're not required to do that animal studies. Yeah, but otherwise, yeah, it's hard. As Dr. Yang said, pick the low-hanging fruit first. I think that's why one of the reasons to pick monoclonal antibodies, pick on something. There's evidence from the class so that we can help to make a decision. I think it will be a gradual step. Yeah. Thank you.
Thank you. Yeah, as a physician scientist myself, I also oversee all the preclinical to clinical, even to approval. So personally, the reason I asked this question, personally, I still feel this announcement is a little bit kind of like for an announcement to do the announcement, kind of like that. I think it's immature. I think it's kind of a little bit too aggressive at this time. Yeah, thank you.
We have an online question. I will read first and then maybe clarify a little bit. Will non-clinical PKTX assay development employee be laid off due to the decision to reduce animal usage? Since there's no way to clarify the question, because this sounds like it's the other way. Because reduce animal usage will promote the assay development. So the question should be asked in the other way. But anyway, I will ask advice from the three speakers. Or I can ask in the other way, what the impact to the assay development company or to the people for this new policy?
I think I think I mentioned PKTK, right? I think at least you need to have some data built on that. If you, I think it's very hard because for pharmacokinetics, right? You're dealing with how our body deal with the drug, right? So like including ADME, everything, right? So sure, you can use some in vitro studies to identify what kind of metabolites they are. I think this is one thing. But for kinetics, I think how I think there are companies developing these assays and check that, but I have no idea how relevant, how accurate it can replace a real animal studies. Yeah, I think, at a later stage, well, you can do PK/PD modeling and help to decide what kind of dose you're going to use your human. Yeah, but I have no clue whether it's good enough and without an animal study with PK studies. Yeah, and there are so many parameters, right? AUC, half-life, what's the correlation when you're using a completely in vitro studies to get those parameters? Yeah.
Thank you. I have a question for Dr. Zhong for the ADC. You have the xenograft model data. The efficacy is excellent. There's a control, there's a treated. So does the tumor come back, or do you have the plan to do that?
Based on the results, it's like the tumor was completely removed. We don't have data like I mean, we don't have further data. This is just a one-time injection.
Thank you. Also question for Shi. So you have a animal compared with the NAM data in one slide. You talk about the cost. One is you mentioned about the monkey, and then the other one is in vitro. Thousands of dollars was millions of dollars. Can you give a specific example, like it provide the similar value, or the NAM has better prediction value than the animal data, or they're comparing side by side for the toxicity endpoint? Is there any example like that?
That's a very good question. Thank you. I think I also feel obligated to explain a little bit. So when I compare the price to the millions to the thousand, I don't mean that the one NAMs can replacing the animal study. I just trying to say that there's some price difference here. So, and also as Dr. Chen mentioned that in the future, if you really want to phasing out animal study, how are you going to use only one organ on chip to replacing everything? So that's my mistake. I shouldn't use that direction. So, but to answer your question, when will be a NAMs will give a better prediction in the animals, I will still use my example using that metabolized parents compounds. So we did try to use an animal study approach to try to answer the question, is this metabolite driven to the cardiotoxicity observed in the animal? But we do not expect to see the cardiotoxicity in the human in the proposed human clinical dose. What we did is that since we have the animal treated with this drug candidate, then we gave that the CYP450 inhibitor. Therefore, we try to prevent the conversion from the parent to the metabolite. In theory, we will have this animal to expose to the more parent instead of the metabolites. However, there's always the devil in the detail. This P450 inhibitor itself has toxicity, which means that we still see the adverse reaction, we still see the histopathology changes, but then that's attributed to the P450 inhibitor instead. So animal model in our hand is not the best model to answer the question. Therefore, we go to the other route. We choose this iPS-derived cardiomyocytes. Therefore, we can just give the parent, give the metabolites, and then see, oh, here it is this metabolites driven to the cardiotoxicity.
Thank you very much. I have a last question for Dr. Chen. Since the FDA policy has a strong interest, and you mentioned there may be a plan to update the guideline, can you give more details regarding which guideline, or is there any timeline regarding which guideline may be updated? Can you provide your advice?
No, yeah, but I'm thinking, at least, they have this publishing this roadmap for this particular topic. I think they should give the industry, right, give everybody a clear clue, what we need. If people want to start in developing more NAMs, what should they do, and what's the criteria, how to standardize that, and before you jump to connect all the NAMs to make a comprehensive predictions and completely replace an animal studies. I don't see it's immediate thing happen, but we need to do step by step and do the qualification for each NAMs and to work together.
Thank you very much. Thank you.
So I think with a lot of discussion with labs, and for machine learning and the AI-based model, those modeling, right? So do you think like it can cover some gaps and integrate the previous experience with previous a lot of clinical data, right? Animal data, and then maybe with labs to help to advance this big goal.
Yeah, sorry. Yes. So maybe I have some thoughts. I think I haven't mentioned that, but I think, then, AI machine learning, I think it may take more time. It's learning the data, what we have already. We need to assemble all this and make a correlation analysis with clinical data, for example, right? And
yeah.
they are more relevant, right, than animal studies, right? Just like the example I have for the chemicals, right? They used 18 tests and compare what's been used and say, well, when you use those tools, it can verify. It's been validated. They came to the same conclusion, right? So you need these steps, right? But then here is, when you have this machine learning tools, for example, FDA has their own tools, right? In-house tools, and EMA may have their own tools, okay? And, how do you know which one is better? Okay,
Yes.
and companies always doing the same thing. So if they have different opinions, which one do you want to listen, right?
Yeah.
Is it like, you have this AdCom, right? You have experts, people vote, right? So is it, okay, so can you vote on toxicities? Do you want to risk that? I think, and then for some real situation, I agree. And then if it's for example, a rare disease, or sometimes the bar should be low, but if it's for a common disease, should you use that, and how do you want to use that tool? Yeah, so I think there should be a lot of debate on that. Yeah, so
Yeah, fully agree.
One last question.
This is more of a comment or follow-up comment on the ADME and NAMs. So if the goal of NAMs is to reduce animal use, there are many ways, not just the NAMs, to reduce animal use. And for ADME, there is microsampling, which you could limit or reduce the animal use, especially for small rodent study, by like 30 to 40% easily. So that's one way. And also the use of NAMs in terms of ADME, they may have better prediction or to aid the selection of animal species. What's the best animal species to use if you're using 3D liver culture or hepatocyte culture compared to the conventional 2D? You might be able to predict the metabolite better. Then you choose a better species to start with. So this way you reduce your kind of a accidental waste of animal because you choose the wrong species to start with. So I think NAMS is not the only way to do that. And that's something we should keep in mind. Animal studies are still of great value. It's not just that animal study is obsolete and everybody should get rid of it. So
thank you.
Thank you, Jack. Dr. Jaya also has FDA experience. With that, we want to conclude our session. And before the next sections that we have the coffee break, we want to thank you. Thanks all the speaker for your hard work and all the audience for your attention. Thank you.